Imagine a world where a simple bacterial infection could become a death sentence. A small wound, a sore throat, even a minor fever, things we barely worry about today, once killed millions of people. But everything changed with one extraordinary molecule, penicillin. But have you ever wondered what actually happens when penicillin enters a bacterial cell? How can such a tiny molecule destroy an entire bacterium? Today, we will dive deep into the molecular battlefield and uncover the fascinating mechanism behind how a beta-lactam antibiotic kills bacteria.
Have you ever imagined that one of the greatest medical discoveries in human history happened by pure accident? In 1928, Alexander Fleming was studying harmful bacteria called Staphylococcus. Day after day, he carefully grew these bacteria in petri dishes, searching for ways to fight deadly infections. Then, before leaving for a vacation, he unknowingly left some dishes behind in his laboratory. When Fleming returned, he noticed something strange, something no one expected. A mold called Penicillium had contaminated one of the dishes. But here's the amazing part. The bacteria surrounding the mold were completely gone, dead. Yet the bacteria farther away were still growing normally. This clear ring around the mold is called the zone of inhibition, an area where bacteria cannot survive because of an antibacterial substance. Fleming realized the mold was producing a powerful bacteria-killing compound. He named this miraculous substance penicillin. Although Fleming could not purify penicillin himself, his accidental discovery changed medicine forever. That single forgotten petri dish eventually led to the first antibiotic, saving millions of lives and transforming modern healthcare forever.
Discovering penicillin was revolutionary, but discovery alone was not enough. The real challenge had only just begun. How could this life-saving medicine be produced in large quantities for the world? Between 1938 and 1941, three brilliant scientists, Howard Florey, Ernst Boris Chain, and Norman Heatley, worked tirelessly to purify penicillin and make mass production possible. Then came another breakthrough. A stronger mold, Penicillium chrysogenum, was discovered, dramatically increasing penicillin production. At the height of World War II, soldiers were dying not only from bullets, but from deadly bacterial infections. Penicillin changed everything. It turned once fatal infections into treatable conditions and saved millions of lives across the world. In 1945, the world honored this extraordinary achievement. Alexander Fleming, Florey, and Chain were awarded the Nobel Prize in Physiology or Medicine.
At first glance, penicillin may look like just a complex chemical drawing, but if we break it into parts, it becomes much easier. Penicillin has three major structural parts. Think of them as three important components working together. The first part is beta-lactam ring, act as the weapon. The most important part is the beta-lactam ring, the heart of penicillin. This small four-membered ring is highly strained, like a compressed spring. Because of this strain, it becomes highly reactive and can easily attack bacterial enzymes. This is the main weapon of penicillin. The second part is thiazolidine ring, act as the support frame. Attached to it is the thiazolidine ring. Think of this as the molecule's support frame. It helps maintain the proper shape of penicillin, so it can fit perfectly into bacterial target enzymes. Without this structure, penicillin cannot bind effectively. The third part is the side chain, or R group. This small part changes how penicillin behaves.
such as its strength, stability, and spectrum against bacteria. The bacterial cell wall is like a protective armor around the bacterium. Think of it as a strong brick wall protecting a house. Without this wall, the bacteria becomes extremely vulnerable. Peptidoglycan is mainly made of a special material called peptidoglycan. Peptidoglycan is like a giant mesh or net made of repeating sugar units. Its two main building blocks are NAG, N-acetylglucosamine, NAM, N-acetylmuramic acid. Now, here comes the interesting part. These NAG and NAM molecules don't just sit side by side. They are connected by tiny protein chains called peptide bridges. These bridges act like cement between bricks, making the wall strong and stable.
So, what exactly does penicillin attack inside bacteria? Does it attack DNA? Does it attack proteins? No. Penicillin has a very specific target. Its lethal targets are special bacterial enzymes called Penicillin Binding Proteins, or PBPs. These enzymes act like tiny molecular builders. Their job is to build and strengthen the bacterial cell wall by creating crosslinks in the peptidoglycan layer. More specifically, penicillin targets an important PBP enzyme called DD-peptidase, also known as transpeptidase. This enzyme performs the final step of cell wall synthesis. It creates the crosslinks that make peptidoglycan strong and stable. But how does penicillin stop this enzyme? Penicillin cleverly mimics the enzyme's natural substrate, called d-ala-d-ala. Because of this structural similarity, the enzyme gets fooled and binds to penicillin instead of the real substrate. In simple words, penicillin tricks the builder, blocks the final wall construction, and weakens the bacterial defense. Now, we learn what chemical reactions actually happen step by step. First, the bacterial enzyme, called DD-peptidase, or PBP, recognizes its natural substrate, d-ala-d-ala. This substrate is essential because the enzyme uses it to build strong crosslinks in the bacterial cell wall. And now comes the clever part. Penicillin mimics d-ala-d-ala, so the enzyme mistakes it for the real substrate and allows it into its active site, like opening the door for an imposter.
After that, it gets inside the active site. A nearby amino acid, usually lysine or tyrosine, activates the serine residue by removing a proton. This converts serine into a powerful chemical attacker. Then, the activated serine attacks the carbonyl carbon of penicillin's β-lactam ring. This is the critical moment where the enzyme begins reacting with the drug. Because the β-lactam ring is highly strained, the attack forces it to snap open. The bond inside the ring breaks, releasing stored strain energy. As the ring opens, penicillin forms a strong covalent bond with the enzyme. The enzyme is now chemically trapped in a penicilloyl enzyme complex. Unlike normal substrates, this complex is extremely stable. Breaking this bond requires enormous energy, creating what scientists call a thermodynamic sink. This makes penicillin a suicide inhibitor. The enzyme uses its own chemistry against itself and becomes permanently inactive. With the enzyme blocked, bacteria cannot cross-link their peptidoglycan cell wall. The wall weakens, internal pressure rises, and eventually the bacterial cell bursts.
Now, this is where penicillin becomes truly fascinating. At first, everything looks easy. Penicillin meets the enzyme, and they bind very quickly. The energy needed for this step is very low, only 3.1 to 3.6 kilocalories per mole. So the enzyme almost welcomes penicillin. But that's exactly the trap. Once binding happens, penicillin forms a strong acyl-enzyme complex. You can see here, the system drops into a deep valley, called the thermodynamic sink. Think of it like falling into a deep pit. Getting into the pit is easy, but climbing out is nearly impossible. For the enzyme to escape, it must go through deacylation. But look at this huge energy mountain, around 53 kilocalories per mole. That barrier is enormous. The enzyme simply cannot overcome it under normal biological conditions. And this is the dramatic moment. The enzyme uses its own catalytic mechanism to react with penicillin, but in doing so, it destroys itself. That is why penicillin is called a suicide inhibitor.
So, what happens after penicillin disables the enzyme? The answer lies in the bacterial cell wall. Normally, bacteria constantly build and repair their cell wall using penicillin binding proteins, or PBPs. These proteins create strong crosslinks in the peptidoglycan layer, giving the wall strength and stability. But once penicillin inactivates PBPs, everything changes. The bacteria can still try to make new cell wall material, but without proper cross-linking, the wall becomes weak, fragile, and full of structural defects. Imagine building a brick wall, but without cement between the bricks. It may stand for a moment, but it cannot stay strong. Now comes the critical part. Inside every bacterium, there is high internal pressure, called turgor pressure. The cell wall normally pushes back against this pressure and protects the cell, but a weakened wall cannot resist anymore. Water rushes into the bacterial cell because of osmotic imbalance. The pressure keeps increasing. The cell swells, stretches, and finally, it bursts. This process is called osmotic lysis. And as if that wasn't enough, penicillin can also activate autolysins, enzymes that begin breaking down the existing cell wall even faster. So the bacterium is attacked from both sides. It cannot build a new wall, and it starts losing the old one. With no protection left, the bacteria dies. Penicillin was once a miracle drug, but bacteria didn't stay helpless for long. They evolved powerful defense mechanisms to survive. First, beta-lactamases. Think of these as bacterial scissors. They cut open penicillin's beta-lactam ring, destroying the antibiotic before it can attack. Next, altered PBPs. Some bacteria change their target proteins, so penicillin can no longer bind properly. This is exactly how dangerous bacteria like methicillin-resistant Staphylococcus aureus resist treatment. Then comes reduced permeability. Some bacteria simply shut the door. They alter membrane channels, preventing penicillin from entering the cell. But wait, there's more. Efflux pumps act like tiny escape machines. Even if penicillin gets inside, these pumps throw it right back out. And finally, the smartest defense? Allosteric control. Some PBPs stay in a closed, protected form, making penicillin almost useless unless a special signal opens them. Amazing, right? Bacteria may be tiny, but their survival strategies are incredibly advanced.
Here's some flashcards showing that we taking earlier. So if this made the topic easier to understand, don't forget to like, subscribe, and share the video. And if anything was confusing, drop your question in the comments. I'd be happy to help you understand it better. See you in the next video.